Howdy, everybody. My name is Kim Hummer, and I'm the research leader at NCGR Corvallis. I just began the recording, so those of us that aren't here at the moment but want to see this presentation a little later will have the opportunity. I'm really pleased to introduce Robert Krueger, my colleague and friend for many years now. I think we're probably the longest standing curators in the clonal germplasm system. I think we were both hired in the 80s or so. And it's exciting to be working, of course, in the positions that we're assigned. We love our germplasm and the NPGS family too. So Robert is in charge of the citrus and date palm collection down at Riverside, California. And I'm going to turn the meeting over to Robert to give his presentation.
Hey, thank you, Kim. And I'd like to also thank you for organizing this. I think this is a really nice little series of get-togethers we're having here. Since we're not able to meet in person very much these days. And it's also telling some of the support staff here, you know, we curators and RLs get to visit the different units, or we used to be able to visit them without too much trouble, you know, at meetings or just whenever, but it's harder for the support staff. So I think it's great that, you know, we're able to organize something so the support staff can see a little bit about our different units. And so I'm trying to put together, hopefully this will work out. We have kind of a hybrid slideshow and live presentation, and we walked through it the other day, but many things can still go wrong and usually do. So I'm going to go ahead and start sharing my screen now. First I have to pull this guy up. And what happened to Zoom? And let's see here. Is it here?
Yeah.
Okay, do you see it, or is it in presentation mode or no? I gotta do that again.
Sorry, I was muted. It's not in, yeah, there you go.
Okay, so I put this presentation together from a bunch of other things with a few new slides. Tell you what's happening at the National Clonal Germplasm Repository for Citrus and Dates. This is our quote-unquote new sign from the year 2000. It was established in 1986, and for the first 15 years, we had a sign that misspelled repository. So we corrected that in 2000, and the sign's been with us ever since. So what's happening right now, there's a lot of construction. On the right, you see my usual view from my office of the Box Springs Mountain and the Big C and the Rose Garden and the variety collection. On the left, you see what's going on right now. We're getting a modular building put in. And so I'm here in the greenhouse area, which is more or less the center of our plant-related activities, but also has a lot less construction noise. So that's a good thing. So this is the famous Peter Bredding slide about our different activities. And I'm mostly going to talk about what we do on the left-hand side of the screen there in order of importance: maintenance, acquisition, regeneration, documentation, and distribution. Mostly about maintenance, acquisition, and distribution. And I'm not going to talk about research at all, except very briefly. And so this is a slide of our staff, sort of. Dr. Mary Lou Pollack, who was our research leader, retired in July. And so that position is vacant. John Priest from Davis, as you guys probably know, is the acting RL. I'm the curator horticulturist, category 4. Manjunath Karamani is a plant pathologist, category 3. We currently have 3 biological science technicians. Vicki Newman and Brittany Moreland work primarily with plants in the headhouse, greenhouse field, etc. Esteban Rodriguez is mostly centered in the lab. We have the secretary, Patricia Moore, and Lee Gross, who is shown in a half-size picture is a half-time maintenance technician. Most of last year, we also had a cooperative university employee Amanda Rostern, and she's gone on to do a PhD program in Florida. So we're very happy for her. Sad to see her go. She's a real good help, but it's good that she's advancing. We also typically employ student employees hired through UCR, and so due to COVID, students are not allowed. Undergraduate students, particularly, are not allowed on campus. So starting in March, we don't have any student help, and that had a big impact on us getting our job done, because things we formerly had these 6 people helping with, now fell to 3 of us usually to get things done. And so 3 of them, Yesenia, Alex, and Bethany, graduated, and so we're able to bring them back as LA appointments. And they've been like a huge help to us to get things caught up compared to what they were in May when we started coming in from being at home most of the time previously. 2 of them, Miguel and Nick, are kind of on hold till they're technically still employed, but they haven't put in any hours since March. So we're waiting to get the clearance for them to come back. Don't know when that will be. So we are located at the University of California, Riverside. And as you can see from my shirt, it was founded in 1907, and we celebrated the centennial in 2007. So it's actually almost as old as I am. And the right-hand picture shows the original citrus, actually, that is the original citrus experiment station. It was actually across town in a different area of Riverside. And it moved to the building that's shown in the picture on the left about 1917. And so we're specifically in an area called agricultural operations. And on this top map, if you can see my pointer, the main campus is over here. Agricultural operations over here. It's about 450 acres total. And we're down in field 12D. You can see our building right there. So it's kind of a cool place. There's advantages to being isolated from the rest of the campus. Disadvantages also, but it's pretty quiet, and it's kind of scenic sometimes. Somebody once told me it's kind of like you have your own private park to work in. And I have kind of a nice picture here of some snowy mountains and some trees. There's even better, because it used to have an antique tractor park there, but by the time I got my camera, it's gone. And I know up at Davis, not too far from the repository, they used to have the antique tractor collection. I one time mentioned that to the former superintendent, Steve Cockerham, and he commented that all their tractors were antiques. So they've actually able to get some new equipment due to their the cooperative agreements we have with them. They get excess government property, so they've got some pretty interesting military-looking equipment now. And so there's our building with our coordinates. And this is our main facilities here. And so we're here in the head house area. Like I said, this is where most of the plant-related work goes on. We have 4 greenhouse chambers here, 6,000 square feet total. A protected greenhouse collection of 16,000 square feet. We have a protected work area. By protected, I mean it's under screen. Lab and office area, and the office trailer. We also use about 7,000 square feet of additional space that we rent from UC Riverside. And we are currently getting the, I showed you the construction picture. This is a modular building, so it will be about 40 by 20 feet wide. And at some point it'll be occupied and serve as additional office space and break room space. In addition, we'll be getting an expansion of our screen house right here. It'll be expanded by a little bit less than 50%. So that'll be a big positive thing, because we're about out of space in the screen house. And so another thing about our location is that in 2017, we're an HLB quarantine zone. So HLB, or one long Bing, is a bacterial disease of citrus, which is very serious, can have a really large detrimental effect on production and even kill trees. And citrus is highly regulated anyway, both by the federal government and state government, but it's even more so when you're in an HLB quarantine zone. And so we actually were not able to do distributions for about 6 months after the quarantine was imposed a couple of years ago. So anyway, and it's had a big even starting before the HOB quarantine, when we had the ACP, Asian citrus psyllid quarantine, it made a big change in how we do business. Because before, if we wanted to go out to the field to collect fruit to extract seeds from, we would typically do it in the morning because it's cooler. Well, can't do that anymore because you can't go in a greenhouse after being potentially exposed to the psyllid vector when you're out in the field. So we have to do all our field work in the afternoon or do nothing but field work, no greenhouse work. So then putting up all these screens has been, you know, a big impact on our daily work. And so here's a picture. I'd hope to be able to walk you guys around the facilities using Zoom on my phone, but the wireless ran out and the cellular was too slow, so you're gonna have to settle for some pictures. And so this on the right is the protected collection I mentioned to you, and on the left, those main greenhouses there. And so starting about 2008, when they first detected the psyllid vector here, we started putting up the screening. And so at that time, APHIS and also CDFA, which is the California Department of Food and Agriculture, started requiring more screen to put up to protect facilities from intaking psyllids. And this is one of these things that probably should have been a good idea even before, but it prompted this kind of existential crisis to you know, prompt a lot of this. It's sort of similar to the situation with the COVID. As you might know, those two vaccines that are probably going to come online first were developed with this novel mRNA technology. And so a large event like that, you know, prompted all this innovative research, you know, concerning vaccine development. And the HOB prompted a lot of innovative research for citrus. Before, we used to have kind of a small little group of citrus people, California, Florida, a few other places. And they've really reached out to people in different locations and different disciplines to really get a lot of new and interesting areas of investigation going. Okay, we also had to screen in our work area. So on the right, you can see what we call the soil card area because it has a soil card in it. And it's there that we do a lot of the transplanting and repotting and this type of thing. And so before it was in the open air, and the screen again protects us. Actually, it's nice because it provides some shade during the summer too. Riverside can get pretty hot during the summer. It's not unusual to get 105, 110 degree days for close to a week or even longer in some cases. So having that shade really helped a lot for the humans as well as the plants. So our holdings, I kind of think of this as a collection of collections. I came up with that phrase in the 1990s. I probably need to change it, find something new and more interesting, but it sort of is. For citrus, we have the citrus variety collection, which is in the field and also in the greenhouse. We also have the protected pathogen collection, which is in the greenhouse solely. The wild relatives are maintained in both field and greenhouse. Some of them never get in the field because they're too sensitive or die for other reasons. And then a few other things that don't really fall into any well-defined group. Date palms are currently maintained at Thermal. We used to have it originally at Brawley, but that group of palms has not been part of USDA for about 5 or 6 years ago now, and is declining terribly last time I drove by a few years ago. So here are numbers, which are probably not accurate anymore. They change, not continuously, but periodically. But we have about 1,600 total accessions of citrus and related species. About 1,200 are in the species citrus. About 300 are in the related species. Now, this probably needs revision because of the taxonomic revisions that Melanie Shorey is working on. So a larger proportion of these will now be included in citrus. The 3 genera that are presented here, Monstera, Fortunella, and Microcitrus, have now been sucked back into citrus. So that group of the relatives is no longer relatives. They're actually the immediate family. We have about 550 pathogen tested accessions in this greenhouse. And we have a total of about 5,600 inventory items or individual trees. And that includes things in the field, in the various greenhouses and screenhouses, and also quarantine items. So the citrus variety collection shown here. And so the origins of this date back to about 1907 at the original citrus experiment station in Riverside. They moved to the current campus location in 1917, and they moved around to different fields over the course of the years, between the last 100 years, really 103 years. So serves as our de facto gene bank, is actually owned by UCR and is cooperative between ARS and UCR. And it's sort of an interesting historical thing. When they were founding these clonal gene banks at the end of the golden era of gene bank establishment back in the 1980s, the ones at the University of California were originally supposed to be run by UC employees. I have in my files some notes taken by the late Professor Jim Cameron, who is a citrus breeder. He went up to Davis for some talk between USDA and USDA and UC about establishing these gene banks both at Riverside and Davis. And so originally they were going to be run through what's now NIFA, used to be CSRRESS or whatever. And at some point they shifted over to ARS, and at that point, it became a de facto ARS resource. But in the case of Riverside, they never completely turned over the materials to us. So we use it cooperatively. We cooperate in decision making about what things to accession and de-accession, and also in some characterization activities. There are about 1,050 accessions in the field, about 2,000 trees. Pathogen status is incompletely known. We have a couple of diseases that are endemic in the area. Citrus rosacea virus, which is spread by an aphid. Stubborn, which is a bacterial caused disease caused by Spiroplasma citri and vectored by leaftoppers. And more recently, HLB is, I think, should be considered endemic in the area because more and more trees and backyards are being diagnosed as having HLB. So far, we haven't detected HLB on the experiment station using the APHIS accepted diagnostics. There are some what they call early detection methodologies that are being used and have found some positives, but they have not been able to be correlated so far with the regulatorily accepted diagnostic. And so mostly because of these disease factors, we distribute only seeds. And currently, APHIS restricts that to accessions within the genus Citrus and Poncirus and our hybrids, which are now back into being citrus. So we can't actually distribute wild relative seeds from the collection at this point. The pathogen tested collection has about 550 accessions and 1,000 trees. The pathogen tested, they meet the APHIS interstate movement requirements, and they also have to be maintained under a compliance agreement, which dictates the structure of the facility, monthly inspections, and different things about the pathogen status. So we retest these annually for CTV and HLB. The current standard calls for every 3 years for protective structures, but we try and do it every year. It's also tested for citrus viroids and cirrhosis at somewhat irregular intervals, and other tests when indicated are possible. Sometimes new pathogens are described, and we will go back and test for things that we didn't know existed previously. And so thus far, none of these quote unquote new pathogens have actually been detected in the screen house. Now I'm going to try and switch over to my phone and see if I can walk around a little bit. If this doesn't work, somebody let me know and I'll stop. So the camera's not switching. My camera's not switching. My camera's not switching.
See you, Robert.
I saw
Going back to plan B and won't take the roving phone to her because you couldn't see what I was seeing, right?
I only saw your face.
Yeah, so for some reason my phone isn't switching, so we'll just sit back down and continue talking here.
We could see, I could see the floor for a little while.
Well, the floor is not really that interesting. I'll let Brittany tinker with it, and if it works later on, then we can always come back to this. John Priest mentioned.
There, I
Priest.
see it.
Okay.
Working?
I think we're walking, Robert.
Okay, you have to unmute that one and I'll mute this one.
Okay, can you hear me?
Yes.
Okay, so Merry Christmas, everybody. So we're approaching this greenhouse here.
We lost you.
As is, the vestibule used to be, now they decided it's not, but we have both the vestibule and the air doors here. So I think so far I'm kind of maintaining. Okay, so this is part of this greenhouse here. You can see the trees here maintained. Here you can't see it really well, but we do have them tagged with accession information. We haven't instituted a barcode yet, and this type of paper was originally recommended by Kim, actually back before I even started. So I started in '94, and they had already utilized this as something to use up at Corvallis. And you can't really see it from here. Not go farther in. The breach is up about halfway down. So now coming back into here. And I think since we got this working, I'm going to continue with this tour and then go back to the slideshow. So this is the greenhouse, and I'm just going to go into the forward portion of it. We have 4 chambers in here, and for this front one, we keep it a medium temperature. This is for propagations of clean material. So these are rootstocks you can kind of see over there. These are some materials we'll talk about in a while. These are some cryo-preserved materials that we're regenerating to see how that goes. And then we got some trees that are propagated, either go in the greenhouse or into the field. The other chambers I won't go into because of the lack of signal. The other ones are mostly for pathogen testing and for quarantine. And so we have a warm room for the warm temperature pathogens and 2 cool rooms for the cool temperatures. So to introduce you to a few more people, you've met Brittany. This is Stevan and Bethany and Lee. Say hi, Lee. So that's that. Here's some trees that are being inoculated. We'll show you inoculations in a little while. And then before I turn this thing off, show you some snowflakes. And this room here is where we do the tissue, not tissue culture, the shoot tip grafting, and also some of the cryopreservation work. And we'll show you that in a minute. So anyway, that's kind of what this looks like. And I think I'm going to go ahead this off now and put back to the, I think we can do the rest of them with this cam, right, Brittany?
This one?
Yeah.
Yes.
Okay, so I'm going to go ahead and leave this now, leave my phone here, go back to the computer.
Okay.
So anyway, getting back to the breach area here, you can see we came in about 8 a.m. on a Monday, and the breach was apparent. This is when we had some really strong winds that were gusting up to about 70 miles an hour, and it came apart at the seams, which is apparently where all these screens come apart. So when we redo this, we're going to have to figure out a better way of doing it. And you see our physical plan on the right. came in almost immediately and started repairing, was still considered a breach. So these approximately 600 plants are no longer available for distribution. So we're going to have to either destroy them or divert them from being germplasm resources and being experimental plants for some of the research projects that I'm not going to really mention to you, but we have several of them that they can be diverted to, that we know they can be diverted to, and there might be other ones too. So Brittany is busy compiling a list of the unique accessions in here that we would have to re-sanitize or obtain from other sources.
Robert, could I ask a question?
Sure.
How old was the screen and how did, so it's just a huge gust of wind or
Yeah,
what?
they're just, the screen is about 5 to 6 years old, and it should last a bit longer than that. But like I say, these screens always separate the seams, because some of the other programs that use these seams or these same screens up in the Central Valley, San Joaquin Valley, they also had a similar breach where it went apart right at the seams. So the way to remedy this, which I have to write a statement of work for, is to, instead of having the seams exposed like that, you cut them so they're the same width as the rafters, and it's secure to the rafters, so there's no exposed seams. And so that'll be more expensive. Last time we did this was about $30,000 to rescreen, if I remember, and this will undoubtedly be more, both due to inflation and to what I just mentioned, the fact that we have to do it a little bit differently. I'm not advancing. What's going on? We just do it this way. Okay, we also have some protected trees that are not quote unquote pathogen tested. So when the psyllid vector of HLB first appeared in 2008, and I got to give credit to Vicki Newman, who was one of the other technicians who's not here now. She said, you know, we should probably have these plants under protection. And that was one of those forehead slapping things, like, of course, we should have done this years ago. So everything that was not represented in the protected pathogen tested collection that was only in the field is propagated and put into these protected structures. So we have about 500 accessions there that are considered unsanitized. Now, we do have some pathogen testing data on them, and in fact, they might actually be quote unquote clean, but they're not sanitized to the current standards that call for therapy, and I'll show you that in a minute. And just briefly, we have a couple of other field plantings. We have some that are right here, citrus research, and here they're planted before being put into the variety collection, so we can evaluate them and see whether they're worth actually making a permanent accession. Then in two different areas, South Coast Research and Extension Center in Irvine, and CVARS, which is the Coachella Valley Agricultural Research Station in Thermal, the low desert. We have both citrus relatives in a case of thermal. We also have a few random citrus plantings. So what are the threats to collection? I already mentioned HLB as far as the citrus threat here. The dates I'll talk about towards the end of the presentation. Fusarium is a threat, and our palm weevils. Human error is also a threat. I don't know whether anybody noticed, but when I went into the greenhouse, there were a couple of dead plants due to emitters coming out. So we keep watching for those things, but you know, errors do creep in. Now, the next line I should actually modify. Government funding. We had a very tight budget in fiscal year 2019 and got a large increase last year. That's what made the construction possible. However, we're always dependent upon you know, Congress and the president to actually give us that, as we will be unemployed as of 2 days from now realize all too well. And then there's university policy. You know, we're considered a valuable member of the university community, but you know, that could change too. Now it's interesting because we didn't have a lease from 2011 to 2019, and we didn't get kicked out. But the lease we have now went from being $1 a year up to about $15,000 a year. And that includes police and fire. That takes up a big portion of our lease monies. Meanwhile, university gave about, I think, 10 acres of pretty valuable land to the California Air Resources Board as a gift. So, you know, you got to stay on the good side, I guess, of the powerful. Anyway, because of these threats, in 2012, we initiated a project with Gail Volk in Fort Collins to do cryopreservation of citrus budwood. And we had a major effort in 2016 to 2017, and we have about 470 cleaning sessions maintained in liquid nitrogen, and we're continuing to work on that. And so we also started some work with date palm in 2018. So using a webcam, this is I don't know if you guys can see this real well.
No, I don't see it at all.
Grab the camera pointer down here, Brandon. So here are some
As far as I can go.
So here we've got some accessions here that have been processed, and these are going to be sent to Fort Collins, Brittany?
Yeah.
So some of them we've processed here and then sent to Fort Collins. In other cases, we just collect the budwood and send it to Fort Collins. And so that's ongoing work. So I'm going to talk about acquisition. And so both citrus and date palms are prohibited by USDA APHIS and need to come in under what we call a PCIP, plant controlled import permit. And so they're also regulated by the state of California. So consequently, we can't just bring something in. We have to go through a whole process of fulfilling the requirements of the PCIP. And that's done by the e-permit system, and you have to submit a protocol as well as your actual application and how to handle it. So there's currently 3 programs importing citrus materials into the U.S. Ourselves, the Citrus Clonal Protection Program was at UCR, and DPI in Florida. And so we and the CCP align pretty closely for reasons I'll show you in a minute. And eventually we want to harmonize everything so we're all kind of on the same page as how we handle material. And so citrus has a center of origin and diversity in Southeastern Asia, which is mostly southern China, northern India, and India, China. And it's moved around the world starting, well, in ancient times, but more recently from the quote-unquote old world to the quote-unquote new world. And so consequently, that's where we have to look for genetic diversity is in these areas. And so rather than just going out and wandering around the forest, we have to actually make an effort to identify and then bring in new accessions. Now, citrus, the main form of propagation is budding onto rootstock. And when we have classes come, they always want to see this, but you guys in clonal repositories are pretty familiar with grafting, so I'm not going to demo it. However, we have what we call graft transmissible diseases of citrus. And so whenever you propagate by grafting, you can potentially transmit that into the grafted plant and get a whole new plant that's infected. And so these are some pictures of Tristeza virus. And this is a historically important virus that in the 1930s and 40s wiped out a lot of citrus down here in Southern California with the quick decline isolates. And that's when this industry started shifting, also with urbanization pressure, shifting from Southern California, so now it's centered up in the Central Valley, about 5 hours north of here. But that just, on the right, you can see this picture of these groves and all the empty spaces from these quick decline strains. Because at that time, citrus planted mostly on sour orange, and sour orange with a sweet orange scion is susceptible to these quick decline strains and they go down really quickly. So you can see how devastating that can be. And on the left are just some of these other symptoms you can see. And so what do we do here? So Dave Gumpf, in the black and white picture, ran the citrus clone protection program starting in the late 70s. And at that time, California had stricter regulations than the federal regulations for citrus. So Beltsville the plant germplasm quarantine office would send it over here to Riverside to be processed and then distributed to the industry. And Dave was also the liaison between USDA and UCR when we established the repository here. And he would always give us a copy of the plants they sanitized to put in their collection. And they began to have capacity issues, and Dave supported and encouraged us to get our own, at that time called a departmental permit, now it's a PCIP. And so, as I said earlier, we try and operate in harmony with them now, and we exchange a lot of materials and a good collaboration. The lower picture shows myself back when I still had a little bit of dark hair, and Georgios Vitolakis, who's the current head of the CCPP, enjoying a Pele Lagarto from southern Mexico, from Tabasco State. And this is the process that we do. And when we receive buds, we actually try and do the same thing with seed source materials nowadays. We seed budwood. It goes into documentation both locally and in GRIN. We do some preliminary testing if possible. And then we try and do an initial shoot tip graft, which I'll talk about in a minute. We also make an initial propagation in case that's not possible. And then if it's initially shoot tipped, it can go to more extensive lab screening. And if you propagate it, then it has to grow for a while, grow through therapy, and then into lab testing. So that'll take, a year or so. After it's rapidly screened in the lab, then we will select some materials for the complete index, which takes 4 to 6 months, after which there is some final lab testing and it can potentially be released. If it tests positive at any step, then it loops back to the therapy step. So some things, for some reason, are easier to clean up than others, and some of them go through pretty easily. Generally, if you can do a successful therapy, then they're clean, but it doesn't always work. And so these are the processes we use. Testing a laboratory is by qPCR or PCR for most pathogens. ELISA is we mostly use for retesting CTV, and culturing is Spiroplasma citrate. Biological indexing is for most pathogens and unknowns, and there are 2 types of therapy: shoot-tip grafting and thermal therapy. And I'm going to mostly give a little bit about these because these are a little bit less known. You guys probably are pretty familiar with these other things. Biological indexing. So a lot of biological indexing was developed here in Riverside over the years, as well as other places. But the basic principle is that we have test plants, positive controls, and negative controls. And so the plant is inoculated, and you look for symptom expression. If the test plants are similar in appearance to the positive control, you conclude they're infected. If they're similar in appearance to the negative control, they conclude they're clean. And so they're redundant to some of the lab tests, which is good. Redundancy is good in testing. Also, it's good for pathogens without sequence data, and in some cases, unknown pathogens. It's also currently required for quarantine release. That could change, but as of now, it's still a requirement. And on the right here, you see, this is one we don't typically do because we do the culturing for stubborn, but this is stubborn, the Spiroplasma citrate and the sweet orange indicator on the left, the healthy, and on the right, the positive control. So you can see, in this case, a pretty dramatic difference. And here are some symptoms. This, I won't go through all of them, but this is cirrhosis, it looks like, tristeza, viroids, cachexia, the stubborn culturing, and that might be HLB. But the thing to remember is you're doing these experiments in controlled conditions where these indicator plants have been shown to show specific symptoms. These don't necessarily have anything to do with what causes the economic problems in the field. And so now we're going to go live, and Brittany's going to show you an inoculation.
I'm going to stop sharing so you can see. So you can see my face and my plant. Okay, let me try to. I can move it down any further. I'll just do it towards the top.
Where is Robert taking us this time?
No, we're sitting right here at the desk. This is a demo at the desk.
Okay.
I just disappeared. Should I do that?
Okay, you have to speak loud though.
So inoculating is basically like grafting, but grafting, except that we don't necessarily need to have a bud. We just need to have the tissue with the vascular system. And so this knife has been pre-sterilized. Typically, we would need to sterilize it with bleach before and after doing this inoculation. So we use a lot of bleach around here, and most of us have clothes that are full of bleach stains. And so we were pretty worried early on in the COVID that we just wouldn't be able to get any bleach for our normal day-to-day plant materials. Fortunately, it seems like bleach is in decent supply at this point. You guys hear me okay?
Probably barely.
And
Wait, where did I put it?
so you can see the what we call the T-cut that she made. And now she's going to take a piece of tissue. This is what we use for inoculating. It's basically like a bud stick. We don't necessarily need the bud.
Kind of hard. I have to do it down here. Sorry. Don't want to cut myself. Okay. I don't think we can see it.
So it's then inserted into the teacup so that the bark of the indicator plant enfolds it. Again, it's similar to do if you were just doing budding.
I don't know if you can really see.
And then it's wrapped with tape, just as we would do if we were doing the budding. So there it is. Thank you, Brittany.
Mm-hmm.
So a few months later, you might have something like this. So this is sweet orange, Madam Vinous sweet orange, just indicator for cirrhosis. And you can see down here where this tape is, this is where the inoculation was made. And here on this plant, you can see the first symptoms are appearing, which is called the shock symptoms. So if you can see this kind of wilted dieback thing there, and also up here, that's beginning symptoms. And so you can see that our test plants and our healthy control, which is the yellow, are not showing these symptoms. And so thus far, the test appears to show that the test plants are negative. But we won't know for sure for about 6 months. So now back to screen sharing. As you guys can tell, like most PhDs, I'm helpless without technicians. So one of the things that this testing program means is that we have to maintain in-plant positives. So we have a collection of actually have a little bit over 100 plants of pathogen positives, and I think we might be the only or one of the only repositories to have pathogens in GRIN.
Oh, we do too.
Okay, there you go. We also need to dispose of material differently. So there's a lab autoclave and a plant blast steamer. However, APHIS is changing some of the rules, so we're going to have to buy a giant autoclave that will have a capacity of close to what this dumpster does, and that's going to cost about $100,000.
Mm.
So some of the constraints for the biological mixing or need for a specific soil mix, greenhouses that can maintain adequate temperature, personnel know what they're doing. So it's time and resource intensive as far as space, time for the plant growth, time for the index, and time to read it. And particularly difficult is the cool temperature testing because, like I say, when it's 105 outside, the swamp coolers can't really keep it below about 90 most of the time. So consequently, we need to do most of our indexing between about October and December. And so I just want to show the soil mix. It's something that's a long time thing we use here in Riverside. It's the UC mix number 3. And with this mixed here on campus. And on the left is Pedro Robles from San de Vegetal in Mexico, and Cosme, who's retired now. Cosme was a good asset because he could explain to the Spanish-speaking visitors in really good detail, everything that went into making this mix. As far as the actual indexing, we typically do 17 to 20 accessions per year. And the cool temperatures are here about 70 degrees, 65 degrees, and the warm temperatures are about 95 and 85, going from centigrade to Fahrenheit. Cool temperatures, we have 6 indicators, typically 4 replicates plus positive and negative controls. Warm temperature, we usually have 1 indicator and 3 replicated test plants with positive and negative controls. So 31 plants per tested accession, or 527 plants total for 17 accessions. One of the major therapy we do here is shoot-tip grafting. This was developed here in Riverside in the 1970s. And it's basically taking a etiolated rootstock, making a tiny cut in it, inserting a apical meristem, and letting it grow again. And here's this one's getting a little big, but you can kind of see the size of the materials that are worked with. And this is all done by young people with keen eyes and steady hands under the microscope. We old people just keep out of the way. That's the best thing to do with the young folk. And so, as I said, this was developed by Luis Navarro from Spain and Chet Roystacker back in 1975 here, writing the paper that won the Pocono Award for horticulture. And here they are in 2019 doing a meeting here in Riverside, the International Organization of Citrus Virologists. That's Luis on the left and Chester on the right. Chester is now 96 years old and he lives across the street from our facility pretty much. So back in pre-COVID days, we used to have coffee with him every once in a while and hear a lot of stories about the old days and how these developments went on I was telling you about. Thermotherapy is pretty simple. It's just heat therapy. The materials are grafted onto these heat-resistant carrizo rootstocks and maintained at 40 degrees day, 30 degrees night for about 4 months. Then if it doesn't kill the tree, then you may have eliminated some of the pathogens. This is better for viral pathogens. There are some pathogens like stubborn, which is a bacterium, also viroids that are more heat tolerant. And so then you for sure have to do the shoot-tip grafting. And this is that same picture you saw earlier. It just shows where we do these different activities. I kind of explained that to you already. Front chamber, plant growth, then the indexing in these back 3 chambers. And our quarantine screenhouse is actually off a site down that direction. So we need to increase our capacity and get into next generation sequencing for pathogen detection. A lot of that's going on in the Vitalochus lab. So they'll tech transfer us at some point. Seasonality, as I mentioned, we have to do this more or less during the winter. And even without indexing, if you go to the NGS, plant growth is a time constraint. We also need to expand our range of pathogens with formal tests. So we're trying to get in new pathogen positives as well as implementing tests for them, laboratory tests. Date palms are also prohibited and come in under the PCIP. In this case, we've got tissue culture date palms, which should be pathogen-free, but they're still requiring testing for phytoplasmas and Kadang viroid. So the phytoplasmas, we've implemented that test, and in the process of, well, we were before COVID, of implementing the Kadang viroid test. And then they added specific species of fusarium, which is kind of complicated. And it's a good idea to add some other fusarium too, which is an important pathogen to date palms worldwide. So I've been in correspondence with people about that. But like I said, it's a little more complicated. So first the Kadang, then move on to fusarium. So all this stuff means there's a lot of permits. I previously had this slide divided into my permits and Mary Lou's permits, but now because she left, they're all my permits. So there's a lot of permits to keep track of. and inspections. We usually distribute 700 to 800 items annually, mostly citrus. Date palms aren't distributed too much except for genetic studies currently. We used to send out a lot more stuff internationally, about 2/3, and then about 10 years ago, due to HLB, about 2/3 of our distributions are domestic, mostly for HLB research and going to all sorts of different states than what we used to send to. We also send to certification programs and nurseries. And so, as you guys probably know, distributions require a lot of documentation on their own, phytosanitary certificates. We usually need a permit for citrus, and dates are highly regulated by most countries, not just the US. Domestically, if we send out material, we need a bedward certificate. Oops. And sometimes I need to write a letter about specific pathogens to go in an AD on the phyto, and occasionally we have to do a non-GNO attestation. I pretty much mentioned this already. We are part of the National Clean Plant Network. So starting about 2009, 2008, something like that, a cooperative effort between NIFA, APHIS, and ARS, established this Clean Plant Network. And so we, as a Clean Plant Center, are part of the network, and we are currently distributing to what they call the minor citrus producing states down here. So even though LSU and Auburn University have centers, they don't have bud source trees like we do. So we're kind of covering this area here. Florida, California, Arizona is kind of one, and Texas have the bigger states, and they have their own programs. These little guys don't. And because of this, we are known also by FAO as being a clean plant center. And so we send to a lot of certification programs in other countries, as well as helping establish a program in Mexico from 2007 to 2013. I was consulting with Sanidad about establishing this National Quarantine and Cleanup Center. It's in the state of Querétaro, it's one of the colonial towns, so it's kind of fun to visit. I'm visitor #1 in her book, so this was like, for me, one of the high points of my otherwise pathetic career. I want to talk about a little bit at the end here about the date palm gene bank. Date palms maintained in thermal. It's about an hour and a half here in what we call the low desert. And so this is actually, well, parts of the Coachella Valley are like over 100 feet below sea level. This is probably about sea level. It's on a slope here. And so during the winter, the Santa Rosa Mountains in the background, these are about 10,000 foot tall mountains, and they'll get a good covering of snow while it'll be 70 degrees down in the desert. So pretty nice. One of these pictures is pretty nice, except of course for these power wires and fences here that I can't Photoshop out. I'm not that good. And so we have about 110 accessions, actually it's a little higher now, 110 accessions of date palms. Date palms are dioecious, so we have both male and female or stamen and pistillate accessions. We have about 500 trees, 700 related species. And it's been very important for genomic studies. We mostly work with a group in Qatar, headed by Joel Malik, and a group in UAE, headed by Michael Peruganan. And they also have worldwide collaborations. And this is in last January, actually this January of 2020, right after PAG, a couple of my UAE group colleagues came out. We had to collect some materials for them. So it's an ongoing collaboration. The reason it's important, this particular collection, so date palms have their center of origin and diversity right around here in Middle East, Iraq, basically. And this is changing a little bit, but this is like a traditional thing. So they spread, starting like thousands of years ago, thousands of years BC, even, through North Africa and through other parts of the Middle East. And this is Mason, Thackeray, and Swingle, some of our early plant explorers. So a lot of our date palm accession have very low PIs, like 7,000. And this is, they wrote about it in these books, which I have some of the original copies in my library about, going to Egypt or Libya or Sudan in like 1903 and having to go in on camels to bring back the vegetative material for propagation. Also, it's pretty interesting. And so the reason that we're important is because a lot of these countries here have had traditional agriculture destroyed. So Iraq, you know, even besides the wars they've had, Saddam had this problem with the Arabs down in this southern marshy area, Basra area, where a lot of date palms were, and he wiped a bunch of them out. So some of the materials we have in the collection no longer reliably exist at their centers of origin. And so it's good for kind of filling in the gaps there with some of these really old varieties. And so our contributions, along with these other contributors, they're now thinking there's actually two centers of domestication, a Western center and Eastern center, or African and Asian, if you want to look at it that way. And you can see here, sometimes there's a little bit of integration from these Eastern populations into the Western ones. And now, I'm told that they haven't published yet, they think there might actually be another domestication event down here. And they also are tracing back, and all the genetics is pretty much beyond me, but they tell me this stuff and I believe it. You know, when did they diverge from into being dioecious plants? So that would be a long time before the domestication, probably. And so that's something I'm still working on. They also identified areas of the genome that are determining the sex of the tree and fruit characteristics and a bunch of other stuff. So it's pretty exciting to be involved in this kind of high-level genomic research. And so lastly, the date palms are really interesting anyway. They're, like I said, they're male and female. They have a lot of cultural inputs. So you go up the tree about 5 times a year to prune, pollinate, thin, tie down, bag, harvest. It's okay when they're a small tree. When they get to be this big, it becomes more of a challenge. And they have these highly specialized guys. The workers are called palmeros. And this guy got out of the bucket and just climbed up into the crown of the tree. I wouldn't do that, even though he is strapped in. I wouldn't trust my bulk to, you know, a piece of webbing between me and the petiole of these leaves here. And then propagation of date palms traditionally is clonal by these offshoots that come off the base of the tree, off of these lateral meristems. And these are removed with this chisel. And so these are made up locally. You can't go down to Ace Hardware and buy a date chisel. This is sucker rod from an oil well, and these are the little things that hold down the rails on the railroad. So they'll weld these together and file them up and everything. And Vince Simmons is now retired. He was the head of the field station out there for their maintain. And he made this slide hammer so you could just slide it and pound it in. They usually just use like a 15-pound sledge and hammer it into the thing and remove it. So it's a pretty labor-intensive thing. It's usually done about May when it's already about 100 degrees out there. So it's, I could say things are different in the desert. And that's about all I have. Thank you.
Oh, Robert, what an excellent presentation. I'm so glad you went into detail on the maintenance of your assigned crops. Some of us are not very familiar with date palms, so that was really exciting to see. I was hoping you'd show us some more also of what I previously called micromalice. It sounds like it, microcitrus. It sounds like you've lumped it into citrus now, or the taxonomists have, but I think
you guys are lucky that we don't have more time, because we have a half-hour video we made of some of the micro-citrus related evaluations we're doing with the citrus variety collection, trying to.
Well, I can see we're going to have a Riverside part 2, perhaps in the springtime. Yeah, thank you so much. I think I'm going to hold off questions because we're just about out of time. And so if people could find their reactions and give some clapping or other signs of thank you, appreciation for Robert's excellent presentation. And
thank you for asking, and I hope the live part went over okay.
oh, it did. It was excellent to be able to walk down and see your collections. So at this point, I will mention that I signed up for a 1:30 presentation for the Corvallis repository. it might be short notice for some of you, and we'll make sure to put the recordings of all of the sessions so far up on the web so that people can see them. And I think after seeing your presentation, Robert, I'm gonna try to do a little revision of mine and do more procedures in our presentation for tomorrow. So I thank everybody, and I hope I can see you tomorrow at 1:30. And I greatly appreciate John Priest and Tracy Matsumoto and Robert Krueger for these excellent 3 presentations that we've just seen. So thank you, folks, and we'll see you tomorrow at about 1:30.
Bye, everybody.